Assalamualaikum class, I'm Nimran Nadeem and I am presenting genetically modified rice, golden rice. First of all, what are golden rice? Golden rice is actually a GMO product which is a biofortified crop and one of the major successes of genetic engineering. A biofortified crop is any crop whose nutritional value has been increased or improved. Golden rice is genetically modified in order to produce beta carotene, which is then metabolized in human body to produce vitamin A, and it is not produced in rice normally. On the right side, you can see the scientific classification of golden rice. Its kingdom, clade, order, family, genus, and species. The scientific name of golden rice is Oryza sativa. Now we will talk about its importance and why it was important to produce Oryza sativa golden rice. The research that led to golden rice was conducted with the goal of helping children who suffer from vitamin A deficiency. Because many children in bad-affected countries rely on rice as a stable food. Genetic modification to make rice produce the vitamin A precursor beta carotene was seen as a simple and less expensive alternative to ongoing vitamin supplements or an increase in the consumption of green vegetables or animal products. In short, the bad affected countries used rice as a staple food, and that is why the rice was modified and to produce beta carotene, and it was a less expensive alternative to other alternatives such as vitamin supplements, green vegetable consumption, and animal products which have vitamin A. Also, golden rice has promised to help prevent millions of deaths and alleviate sufferings of children, and it can help in combating VAT and micronutrient malnutrition in developing countries. As we are talking about vitamin A deficiency, we first should know what is vitamin A and what it does in our body. Vitamin A, also known as retinol, is an essential vitamin and it is used in retina to create pigment. Pigment is created in the rods and cones of the eyes, and therefore it is important to promote good day and night vision. Vitamin A deficiency definitely can cause visual impairment because it is related to retina and uh, rods and cones of our eyes, which can ultimately lead to blindness. It can also cause keratinization of the mucous membranes of the soft tissue, where keratinization in normally should not occur. But in soft tissues, it can cause keratinization, such as in the lungs and GI and urinary tract. In 2005, 190 million children and 19 million pregnant women in 122 countries were estimated to be affected by VAT. VAT is also responsible for 1 to 2 million deaths, 500,000 cases of irreversible blindness, and millions of cases of xerophthalmia, which is drying of the eyes annually. And therefore, it is important to produce golden rice to combat this situation. Here you can see the countries which were affected. This is a graphical representation of the countries which were affected by bad. The red color shows the most affected countries, orange and then orange, then yellow, and then green, and the dark green color shows the least affected countries, and the blue ones are where no data is available. And you can see most of the countries in the world are affected by that, and that is why it was important to produce golden rice to combat this situation. Now we will talk about the biosynthesis pathway, how golden rice was produced. Golden rice was created by transforming rice with two beta carotene biosynthesis genes. The two genes are phytoin synthase (PSY) and phytoin desaturase (CRT1). The PSY and CRT1 genes were transferred into the rice nuclear genome and placed under the control of an endosperm specific promoter so that they are only expressed in the endosperm. Generally, the leaves of the rice have this um, beta carotene, but because it is not present in the endosperm, it is not expressed in the rice. That is why it is first it is first placed under the control of an endosperm specific promoter, and then it is expressed in the endosperm. The bacterial CRT1 gene was an important inclusion to complete the pathway, since it can catalyze multiple steps in the synthesis of lycopene. The end product of the engineered pathway is lycopene, but if the plant accumulated lycopene, the rice would be red. Then why these rice are yellow in color? The recent analysis has shown enzymes process the lycopene to beta carotene in the endosperm, giving the rice the distinctive yellow color for which it is named. The original golden rice was called CGR1. This is the pathway, a simplified overview of the keratinide biosynthesis pathway. The geranyl diphosphate is already present in the rice, and the enzymes expressed in the endosperm of golden rice are shown in red, which catalyze the biosynthesis of beta carotene from geranyl diphosphate to lycopene. This lycopene is then converted into beta carotene, and alpha carotene and beta carotene is the product which we want. This beta carotene, when is present in the human body, is converted into retinol, that is vitamin A. Now comes golden rice 2. In 2005, golden rice 2 was announced, which produces up to 23 times as much beta carotene as the original golden rice. Okay, why golden rice 2 was needed is because you can see in the picture. This is the wild. The first one shows the wild type, and the second one shows us the golden rice one. Golden rice one is a bit um, pale colored because it have less beta carotene. By bioanalysis, we concluded that the golden rice one had less amount of beta carotene, which was needed to combat fat. That is why golden rice 2 was produced in 2005, so that enough beta carotene can be produced in the body. They combined the phytoin synthase gene from maize with CRT1 from the original golden rice, so that this golden rice would have enough of beta carotene which can produce retinol in the body. And this is all about golden rice. Thank you.